Olá, Eduardo, tudo
Oi,
bem?
tudo bem, Rahel? Eu
Tá
acho,
tudo funcionando.
acho que já estamos online, inclusive, eu estou
Olá,
aprendendo a
bem?
operar aqui. Eu
Tá
acho,
tudo funcionando.
acho que tem um erro. Estamos online, inclusive, estou aprendendo a...
Você tá no mute.
Estou tentando tirar, eu estava com várias janelas abertas aqui,
Ah,
e eu acho que ele está recebendo numa e transmitindo em outra. Mas vamos tentar aqui, eu estou achando todas as janelas abertas e mas deu certo. E eu vou abrir em outra máquina,
tá.
que aqui está muito perto a outra que estava testando a recepção aqui. Mas eu acho que está funcionando. Eu vou abrir o YouTube em outra máquina, que eu vou tirar meu vídeo, tá? Mas parece que
Ótimo.
está funcionando.
Tá, eu vou já tentar compartilhar minha tela e tudo para ter
Deixa
certeza que
eu
tá tudo funcionando.
colocar como co-host. Aí pode já compartilhar. Então, o Zoom está funcionando bem. Só vou verificar o YouTube para ver se está transmitindo.
Tá. Aí.
Está funcionando bem, então.
Tá
Estou
dando para
vendo
ver
bem.
meu slide?
Tá. Mouse, tá bom.
Ótimo.
Ótimo. Eu vou, então, em outra, não saí da minha sala ainda, eu vou ali no outro computador só para testar o que que a gente enxerga pelo YouTube, tá?
Beleza.
Mas parece que tá tudo certo aqui. Oi, pessoal, tudo bem? Bom dia aos que estão no horário do Brasil.
Bom dia.
Boa tarde aos demais horários.
Bom dia.
Então, já estamos com vários online, vamos esperar as 10 horas para dar início, mas a gente fez alguns testes e parece que está tudo certo com a transmissão via YouTube. É a primeira vez que a gente faz isso ao vivo, com Zoom e YouTube simultâneos, e pelo jeito funcionou a configuração aqui, está tudo ok. Então, vou dar uma apresentação oficial, né? Mas temos uns professores e alunos já entrando via Zoom, então a gente vai ter as duas opções. A real tem o pessoal do programa vai entrar pelo Zoom e o pessoal os outros participantes vão entrar via YouTube. E tem as duas possibilidades. Então, no momento, já temos alguns no YouTube aqui. E no Zoom, temos aqui a professora Laura, que está me ajudando a organizar esses seminários internacionais nesse semestre, o professor Júlio, que trabalha com ecologia de primatas, o professor Renato, que trabalha com aracnologia, tinha mais um ou dois
Nelson,
professores, que o
o
professor
Nelson.
Nelson Fontoura, que trabalha com ecologia aquática, então vários professores, alunos do programa estão entrando. Você quer testar o teu áudio, Rahel?
Pode ser. Tá dando para ouvir tudo?
Testa novamente. Agora que eu acho que o meu áudio está muito baixo.
E agora?
Deu, não, tudo
Tá.
bem. Tá bom.
Ótimo.
Então, pelo meu relógio são 10 horas. Podemos esperar mais um minuto. Assim. O Pedro Ferreira entrou também, que trabalha bastante com análises ecológicas. Bom dia, Carmen. Esperamos mais um minuto, mas eu acho que podemos iniciar. O que vocês acham? Temos
Acho que sim, já são
16
10
16 no Zoom, alguns mais
Certeza.
no YouTube. Então, bom dia a todos, né, aqui no horário brasileiro. Boa tarde, Rahel. E, né, alunos que estão em outros fusos horários, tem alguns que estão. Então, essa é mais uma edição do nosso Seminário Internacional em Biodiversidade, da PUC-RS, né, uma promoção do nosso programa de pós-graduação em Ecologia e Evolução da Biodiversidade. E dessa vez, pela primeira vez, tentando, né, transmitir pelo Zoom para o público do programa e pelo YouTube, pelo nosso canal simultaneamente. Estamos vendo que funciona, então isso já é uma boa notícia. Nesse mês, então, temos o prazer de receber a doutora Rahel Sollmann, ou Rahel, deve ser a pronúncia mais correta. A Rahel é alemã, mas passou muito tempo no Brasil, fez vários estudos no Brasil, tem uma experiência internacional bastante grande e atualmente é pesquisadora do Instituto Leibniz de Zoo and Wildlife Research em Berlim, na Alemanha. Desenvolve vários estudos interessantes com diferentes sistemas, né, com modelos estatísticos em ecologia de vida selvagem. Então, a gente conversou em Berlim há vários meses e ela manifestou interesse e disponibilidade em dar um seminário para o nosso programa, o que muito nos honra. Ela vai dar o seminário em inglês, mas ela fala muito bem português, né? Então, até tive que perguntar se ela ia falar em inglês ou português, de tão bom que é o português dela. Então, fiquem à vontade depois para fazer perguntas, tanto em inglês como em português. A pergunta pode ser feita né, verbalmente, seria legal os alunos perguntarem, pelo Zoom mesmo. Podem mandar pelo chat, que a gente ajuda, e eu vou tentar ver se pelo YouTube vem perguntas. Isso eu nunca tentei fazer, então se eu conseguir ver como que se enxerga as perguntas do YouTube, eu vou tentar direcioná-las também. Tá? Então, é um prazer receber a doutora Rahel. Vou trocar para o inglês, então, Rahel, thank you very much, and we can switch to English. And it's a pleasure to have you here as our speaker for this month's international seminar. And I think your projection is working, and everything seems to be working, so we can get started.
Excellent. Muito obrigada, Eduardo. Boa tarde, bom dia a todos. Thank you very much for inviting me to give a seminar. The honor's all mine. So yeah, today I will talk about hierarchical statistical models in wildlife ecology, but I will keep the statistics to an absolute minimum and just focus on the concepts. If at any point you stop hearing me or anything goes wrong, please just let me know and I can stop. So many questions in wildlife ecology, conservation, and management revolve around quantities such as population size and dynamics, species distribution or habitat use, or species richness and diversity. And these quantities essentially all relate to a very simple question, namely, how many animals or species are there and where are they? Now, while that question might seem trivial, it can actually be very difficult to answer. And the big problem is that that it can actually be very difficult to enumerate or detect wildlife. I'm going to give you a little example of what I mean. Here are 3 pictures of cats, and I'm going to give you a second to find the cat in each picture. So in case you haven't spotted them yet, there is a Pallas cat over here. There is a snow leopard blending in with its surroundings over here. And here's my cat being an expert hider over here. And so what this illustrates is that even in broad daylight, wildlife can be very elusive and hide in plain sight, and we might fail to detect it even though it's there. So we're not very good at detecting wildlife. And what's possibly even more problematic is that our ability to detect wildlife can vary with the method we're using, the species we're studying, the habitat we're studying animals in, and so forth. So for example, it is a lot easier to see a large mammal that lives in open habitat compared to a small carnivore that likes to hide away. So when we're going to the field and we're trying to measure ecological quantities or processes of interest, then unfortunately, typically, we can only observe those imperfectly, and the level of imperfection varies. And today, I want to talk about hierarchical statistical models as a tool to deal with that problem by jointly modeling both an ecological and an observation process. And I'm going to start out just talking about hierarchical models in general, more specifically of how we can use that framework to address the problem of imperfect detection. Then I'll focus most of my time on 2 specific modeling frameworks. I'll talk a lot about occupancy models that are used to estimate species occurrence, and spatial capture recapture models that are used to estimate abundance, density, and spatial distribution. And then I'll give you a brief general outlook. So hierarchical models are a very broad framework, and fundamentally, they just describe a model that has multiple levels, in which one level is conditioned on another level. Conditioned just means it depends on another level. So here's a simple example of what that could look like. Imagine we're measuring something, let's say species diversity, and we're measuring that in a landscape. In that landscape, we have multiple patches of habitat, and within each patch, we have multiple sampling locations. Well, then particular patch level attributes or variables may affect species diversity, such as patch size or patch isolation. And now if within a patch we have multiple sampling locations, then the things we can measure at these multiple sampling locations are all affected by the same patch characteristics, by the same values of that variable X. And so what we can measure at a given point in here depends on which patch that point is located in. So we have a hierarchy here where the point level process depends on the patch level process. But today I want to focus on hierarchical models as a tool to disentangle a different kind of hierarchy, namely that of an ecological state, such as species occurrence, abundance, or distribution, and conditional on that, an observation process that generates our data, such as species detections or counts of individuals or individual detections. And as I already said, these observations are typically imperfect representations of the true ecological state due to imperfect detection. Now, I want to briefly mention that there are many different observation processes, and imperfect detection is just one of them. But it is really pervasive in wildlife surveys, and that's why I want to focus on this particular observation problem today. So how does imperfect detection work more formally? Let's imagine the simplest ecological state. We go to a location, we want to know whether the species is present, we'll denote that with a 1, or whether the species is absent, we'll denote that with a 0. Now we can go to locations and try to determine species presence or absence. And there are multiple things we can observe. So at a site where the species is truly absent, we can never observe the species unless we lie, but let's try and stick with good scientific conduct here. So if the species is absent, we can't detect it. But at a site where the species is present, two things can happen when we go out and try to survey that species. We can be successful and detect the species. In that case, our observation is a 1, or we can fail to detect the species. In that case, our observation is 0. So this illustrates how our observation process is conditional on or depends on the ecological state, because the ecological state dictates what we can observe. It also illustrates that the ecological state is partially latent, because imagine in a real survey situation, we don't know what that true state is. That's the whole reason we're going out and sampling. So now if we go out and we see the species, then we know for sure it's there. But if we go out and we don't see the species, that's the time when we really have to figure out, well, is the species truly absent, or did we just fail to detect it? And so in that sense, our true ecological state is partially latent. That concept of imperfect detection applies to other ecological states as well. So take, for example, the ecological state of abundance. Let's say there's a population of 5 squirrels in a location, and we now go out to count how many squirrels there are. Well, then in most cases, we will count fewer individuals than are actually out there. For example, we may count 3 squirrels of those 5. And again, the ecological state here determines what we can observe, because our observations are limited by true abundance, right? In this case of 5 squirrels, the most we can observe is 5 squirrels. So again, we have a conditioning of our observation on the true ecological state. And so more broadly, we can define our observations, the data we actually collect in the field, as being a function of the true ecological state and some parameter that's typically called detection probability, denoted with P. And then depending on what the true state is that we're trying to sample, what P means can be slightly different. If we're trying to determine whether a species presence, then P is the probability of observing the species given that it's there. If we're trying to determine abundance, then P is the proportion of individuals we observe. And the big problem is that we don't know what that P is before we go and do a study. We actually need to estimate detection probability from the data that we collect in order to study the ecological state. And in order to do that, we both need to collect data in a particular way, but we also need to use particular statistical models. Why do we care about imperfect detection? Well, if imperfect detection is not accounted for, we typically underestimate the quantity, the ecological quantity we're interested in. So for example, here are 2 plots that show the distribution of a dragonfly throughout Switzerland. And on the right-hand side, we have a model that does not account for imperfect detection. On the left-hand side, we have a model that does account for imperfect detection. And I should say the darker the color, the more likely the species is to occur in that location. And so you can see that if we ignore imperfect detection, we hugely underestimate how likely the species is to be present throughout much of Switzerland in this case. And if detection is not only imperfect, but also variable, and we don't account for it, well, then we can actually make the wrong inference about what kind of variables our ecological state is correlated with. So without giving you too much detail, we ran an example analysis on photographic camera trap data of this small felid here. It's leopard cat that we sampled in 3 study areas in Borneo, and we constructed an abundance index that does not account for detection. And you can see on this plot here, I've plotted the abundance index for the 3 study areas, and it appears that the species is equally abundant in all 3 study areas. But when you use a method that properly accounts for detection probability and the fact that it may vary between the study areas, then a different picture emerges where the species, there's actually a clear gradient in abundance or density across those 3 study areas. That's something we would have completely missed if we didn't account for imperfect and varying detection. So then let's start talking about how we can disentangle this problem of imperfectly observing the things we're typically interested in. And we're going to start with the probably simplest hierarchical model that deals with the true state being species presence. So this is the same graph we saw earlier. The species can either be present or absent, and depending on whether it's present or absent, we can either observe it or fail to observe it. Okay, so how do we need to adjust this situation to be able to deal with imperfect detection? Well, first, we have to determine the ecological state. So we have to survey at a collection of sites or sampling locations. We'll index them with J. And then at each of these sampling locations, we also have to implement repeated observations. So we have to go to each of these locations multiple times. And our data then looks something like this. At each location, we have multiple observations. And so now again, at a location where the species isn't present, we can never observe it. So our data will always look like this. There will always be all zeros. But at a site where the species is present, our data could look something like this. This is just one example. So in this example, we detected the species in the first visit, but we failed to detect it in visit 2 and 3. And so here's one very important thing. We need to be able to assume that the true state does not change across a repeated visit. So in other words, if the species is truly present on our first visit, it also has to be truly present in our last visit. What that means for study design is that these repeated visits have to happen over a fairly short time frame. That'll depend on the ecology of the species. But if we can make this assumption, then these repeated data can now give us information about the detection probability. So you can look at this and think about it. We went to the site 3 times because we saw the species in the first visit. We know it's there the whole time because we can make what is called the closure assumption. Well, then we know now that visit 2 and visit 3 were actually failures to detect the species. And so in this little example, we would have a 1 in 3 chance of detecting the species if it's truly present. That's the math behind it, or the statistics behind it are a little more involved, but that's the general idea of how these repeated data can give us information about detection probability. So in occupancy models, then what we're trying to determine is the ecological state, that is whether the species occurs at a collection of sites. More specifically, these models estimate the probability that a species occupies a site. And we can also answer the question which variable influence that probability of occupancy. And on the observation level, where we have the species detection, non-detection data repeated over several sampling conditions, we ask whether conditional on being present, what is the probability that we can detect the species? And also on this level, we can include variables that explain how probability of detection may vary in space or time. And I want to give you an example of using an occupancy model to address a biodiversity conservation question. This is work from my former grad student, Daniel Rocha, in Brazil. give me 1 second. I have that the hiding cat that you saw in one of the early sites is sitting in front of my monitor. So I'll just get rid of him. Here he is. Sorry for that. Okay, so back to occupancy modeling. So as you're probably all aware, the processes of climate change and land use change are threatening to convert large tracts of the Amazon rainforest to a more open savanna-like habitat type. And Danielle, my former grad student, was interested in how this will affect Amazonian mammals, more specifically their space use. And in order to answer that question, he set up multiple camera trap surveys in 4 protected areas in the southern Brazilian Amazon. So here you can see the location of these protected areas, and then a zoom in where all these blue symbols are camera traps. And what you can see in these protected areas, there are 2 predominant habitat types. The green is forest. And because we're in the ecotone between the Amazon forest and the Cerrado savanna here, there are actually naturally occurring savanna patches in these areas. And so the habitat looks something like that. And what that allows us to do is that we can see how these animals react to these different types of habitats without the confounding effects of human influence. And so his specific research questions were, how does the space use of species respond to savanna habitat at 2 different scales? First, at the local scale, so the immediate habitat that a camera trap was located in, that was either savanna, riparian forest, or continuous forest. And then we also looked at a slightly larger scale because animals often respond to sort of the habitat composition around a location. So we looked at the percent savanna at a 100-meter circle around each camera trap. And then the second question was whether that response to savanna habitat depended on the species' global distribution. So out of the 31 species that he detected in his study, some are associated in their global distribution or range-wide distribution predominantly with the Amazon basin, such as the short-eared dog. There were some species that are predominantly associated with the Cerrado, such as the maned wolf, and there were many species with wide distributions that cover both types of biomes, such as the collared peccary. Why would we use occupancy modeling to answer that question? Why not just look at where these animals were photographed? Well, first off, camera trapping may miss species altogether. We want to account for that. But how detectable species are also depends on their size. Larger species are typically easier to detect. And if we want to compare species, then we need to account for that. Detectability also depends on the location. So I put an illustrating picture down here where you see a camera trap photo taken in forested environment where it's quite easy to find trails or openings that are likely to be used by animals. And so you have a really good chance of photographing animals there. But when you're out in the open Cerrado grasslands, it is much harder to target places where animals move. And so the detectability can really depend on habitat type. And it finally depends on sampling effort because cameras sometimes malfunction and so forth. And so we can take all of this into account by putting it into the observation part of our occupancy model and thereby get estimates of species occupancy that aren't biased by these processes. So what did we find? What were species relationships with savanna habitat? I'm going to walk you through these result plots. On the x-axis, you have the effect size of a camera trap being in savanna habitat. So what that means is if the effect is positive, then the species was more likely to use a location in savanna habitat relative to forest. If the effect is negative, a species was less likely to use savanna habitat, or other way around, it was much more likely to use continuous forest habitat compared to savanna. The species are grouped into these 3 groups that I presented to you. The top 4 species are the Cerrado species, the bottom species are the Amazon species, and that big chunk of species in the middle are the generalist species that occur in both biomes. And so this is the local effect of savanna, so the immediate habitat type. And what you can see is, as expected, the Cerrado species tend to respond positively. They like savanna habitat. The Amazon species down here tend to respond negatively. So again, as expected, they clearly favor forest. A little bit other than we expected, all of the generalist species also tended to prefer forest. Some of them even strongly prefer forest. So that was on the local scale. And then we can look at the 100-meter scale, where we looked at the percent savanna habitat in a 100-meter buffer. We did that separately for each of the 3 immediate habitat types. And without going into too much detail, again, you can see that if the camera is located in savanna habitat, the Cerrado species don't really care. Actually, they don't really care all that much about what's in the surroundings. The Amazon species, they tend to respond negatively to savanna habitat in the larger scale, no matter where the camera is located, but most strongly if you're in the savanna habitat. And then the generalist species, again, also respond negatively to savanna habitat at the larger scale, particularly when you're immediately in the savanna. So what did we conclude from that? We by no means want to imply that a species like, for example, a puma cannot use savanna habitat, but we do think that these results indicate that most mammals, even those that are generalists, will lose access to the habitat that they prefer under conversion of rainforest to savanna habitat, and that can have negative implications for their fitness, for their physiology, and so forth. And so this, in the long term, may hamper the ability of protected areas to safeguard populations of all these species, especially when you think about the southern edge of the Brazilian Amazon. It's a landscape incredibly affected by forest fragmentation. And so protected areas are really important to safeguard populations of these large mammals. And we looked at the effect of natural savanna habitat, and that was already negative for so many species. So the effects will likely be much more negative when we look at non-protected, non-natural savanna habitats, where wild animals will also have to compete with domestic animals, deal with hunting, and so forth. But we can use occupancy models for more than just looking at how species space use is affected by predictors. So remember that occupancy models estimate how likely a species is to occupy a particular location. Well, we can use that information to calculate the percent area occupied by a species. So it's the proportion of the area of our study area that's occupied by a given species. It's also abbreviated as PAO. And we can use that as a measure of prevalence. So here's just an example. If this brown blob is our study area and the green one indicates the distribution of a species within our study area, then a small PAO would mean we have a rare species, and a large PAO would indicate a common species. If we do that for all species in a community of species, so say for all terrestrial mammals, well, we then calculate the relative prevalence and can use that to start looking at community composition. I'm sure you've all seen these kinds of plots. Typically, they use relative abundance to make these plots, but we can build them based on relative prevalence. So here's a toy community of 4 species with one really common species, dominant species, an intermediate species, and 2 rarer species. And from there, once we have relative prevalence, I should say that prevalence is much easier to estimate than abundance, which we will see when we talk in the second part of my seminar, when I talk a little bit about abundance estimation. So we can take relative prevalence to go beyond just these simple community composition plots and actually look at species diversity using diversity profiles. A diversity profile is this thing you see down here. It is a calculation and a visualization of diversity along a gradient Q of how much rare species contribute to diversity. So when Q is 0, all species contribute the same to diversity, no matter how rare or abundant they are. So basically, we just end up with species richness at Q equals 0. But as Q increases, rarer species contribute less and less to diversity. And so if you see this profile go down really steeply, that means that your community is dominated by few common species, that it's a very uneven community. So at larger Q values, diversity takes into account both richness and evenness of the community. Here's just a quick little example of building such diversity profiles for 4 different communities of birds in Borneo. These profiles are built based on occupancy model output. And you can see that 3 of these communities have very similar species numbers. Their richness is almost identical. But the top 2, the black and the blue line, which is continuous forest habitat and riparian forest habitat, are much more even. So there aren't really that many dominant species in this community compared to the green habitat, which are isolated remnants of riparian forest, so fragmented riparian forest. And then this, in contrast to riparian forest that's situated in oil palm plantations, where overall bird diversity is a lot lower. So you can learn about how particular things affect community level diversity based on occupancy model output. So to summarize, occupancy models have been incredibly popular. They're widely used for a variety of things, and that is most likely because the data, they're so flexible in terms of the data they can analyze. All you need is species level detection, non-detection data, and you can collect that with direct observations or camera traps or other remote recorders. You can use signs or scats to detect species presence. You can use more novel tools such as eDNA. The models themselves are also endlessly adaptable. You can build community models. You can look at occupancy dynamics over time. You can start studying species interactions. And as I briefly showed you, you can use the output from these models to look at more community level questions as well. The major drawback of these models is that occurrence is a very coarse measure, right? It only tells you whether the species is there or not. And in many cases, that may not be enough to answer the questions we have or to appropriately protect species. And if we need a more fine-scale measure, then we're going to want to estimate abundance. And so I now want to talk a little bit about hierarchical models for abundance estimation. There are two large classes for abundance estimation. The first class is that of count-based models. And in count-based models, I already showed you this general concept. We typically observe fewer species than there are. And fundamentally, the true abundance is the number we observe divided by detection probability. So we need some additional information to estimate that detection probability so that we can get from our counts to true abundance. And there are a variety of approaches to estimate detection probability. You can implement repeated visits similar to occupancy modeling. There's distance sampling. There's double observer sampling. Many, many different methods that I'm not going to get into today for time reasons. Instead, I'm going to jump straight to what's considered the gold standard of population estimation and the other large class of abundance estimation there is, and that is the huge framework of capture mark recapture modeling. The big difference to count-based abundance models is that in capture mark recapture, it's not enough to count individuals. You now have to be able to tell them apart so you can recognize individuals repeatedly. That can be done based on natural marks, such as stripe or spot patterns. In case of naturally unmarked species, that can also be done with artificial marks. If we have a population of individually recognizable animals and we sample from that population repeatedly, then we can build, then we have repeated individual level detection data, which are also called encounter histories. So that's what encounter histories look like. In this table, it's one row of data for each individual that we detect. And then for each of the repeated times we sample the population, we note whether or not that individual was detected. Similar to occupancy modeling, these models assume population closure. So that means over the repeated visits, no individuals are recruited or lost. And if that condition is fulfilled, well, then these kinds of data provide, again, information about the probability of detecting an individual, and that then allows us to estimate true abundance. The logic is the same here as for occupancy models. If you look at that individual 2, for example, it was detected in occasion 1 and occasion 3. It was not detected in occasion 2, but because we can assume population closure, we know that in this case here, we actually failed to detect the individual because we know it was there. And so we can, you know, again, the logic is, okay, we can see that in 2 out of 3 attempts, we detected the individual. So that gives us information about detection probability. But these kinds of capture-recapture models have a big drawback. So this is what the data look like. But in reality, this is typically what sampling looks like. We will have, in most cases, we will have a number of detectors here, these black dots out in the field. These could be camera traps, these could be life traps, whatever you use to sample your population. And then we will have animals. These are these colored blobs here. And these animals live in particular locations and move around. And as they move around, they interact with different traps. Well, the capture-recapture data we've seen so far completely ignores the spatial nature, both of populations, so individuals, and of sampling. And that has several negative implications for estimates of abundance. Again, for time reasons, I'm not going to go into the details. Suffice to say, it can lead to big bias in population estimates. And so there's a relatively new framework, and I say relative because by now it's been around for almost 20 years, which is crazy, and that is spatial capture-recapture. And in spatial capture-recapture, instead of condensing the data down to just each individual and each occasion, we create these individual encounter histories for each trap in our trap array. So if they were camera traps, we'd say, okay, at camera trap 1, individuals 1, 2, 3, and so forth were detected at that trap in sampling occasion 1, 2, 3, yes or no. So we retain that spatial information on where individuals were detected. Well, if we retain that spatial information, or if we're interested in that spatial dimension of sampling and the population, then we also have a more complicated ecological state model. Remember, before the ecological state was just abundance. Now it's a little more complicated. Now our ecological state also has to deal with the spatial locations of individuals. And so to do so, we assume that each individual, so the red dots here are hypothetical individuals in the population we're sampling with these black dots. So we assume that each individual has an activity center, and we have some information on where that activity center is because we know where we detected an individual. In the simplest case, the model assumes that activity centers are distributed homogeneously across the study area. And abundance is defined as the number of activity centers in our study area. Well, now that we have space in this model, we also can describe the detection model in terms of space. Remember, before the detection process was just, is the individual detected, yes or no? There's one probability for that to happen. Well, now we can actually formulate a model that describes detection probability of an individual at a given trap as a function of the distance between that individual's activity center and that trap. So we can look at this in two ways. So here's one little plot. As the distance to an individual's activity center increases, its detection probability decreases. Or you can look at it over here, where if this is an individual activity center, then the individual will likely be detected at these traps that are close to its activity center, but the further away you get, the less likely you are to detect that individual. And these functions are governed by detection parameters. So for example, in a simple case, we have some baseline detection that tells you how detectable an individual is if a trap was right at its activity center. And we have a scale parameter that determines how fast that decline happens, and that is related to how far animals move. And so here is a very brief example from my PhD work in Brazil, in Emas National Park, where we set up camera traps all across the park. So here, that's a map of the national park. In yellow, you have grasslands. In green, you have forests. All these yellow circles are camera traps. The black lines are roads. So we set camera traps throughout the park. They were out for 3 months of sampling. And in that time, we detected 10 jaguars. 5 were males, 5 were females. But you can tell by the number of pictures that we recorded the males much more often than the females. So clearly, there's variation in detection probability between males and females. We also found that the cameras on the roads detected jaguars much more often than the off-road cameras. So clearly there's also spatial variation in detection probability due to where the camera was set up. And so in the spatial capture framework, we were able to build a model that accounts for all of that. It accounts for the fact that males seem to move more and larger distances, and that jaguars in general like to walk along roads. And with all of these sources of heterogeneity accounted for, we arrived at a very low density estimate of jaguars for Emas National Park. So the case study in Emas used this sort of simplest case that I introduced before, where activity centers are distributed homogeneously across the entire study area. In other words, that means that we expect density to be the same everywhere in our study area. Now, of course, that is unrealistic when we have large study areas that vary in habitat when they're heterogeneous. And it may also explicitly be our interest to see how density varies as a function of habitat, because variation in density is a second order habitat selection. So that's often of interest in ecology. And we can expand these simplest spatial capture recapture models to spatial capture recapture models that allow for inhomogeneous density. So in those models, density varies as a function of a covariate. So imagine these squares here. This square is our study area, and the colors indicate a covariate value. Now we can, and density varies as a function of that covariate. So you see density is higher in areas where the covariate is high. Well, then this relationship between density and the covariate can actually be estimated directly inside the spatial capture recapture model, without any additional information on animal movement or anything. And so I want to give you a short example of that kind of modeling. That's not from Brazil, that's now from North America, studying northern flying squirrels in California, that are those adorable little furry creatures. So northern flying squirrels are considered old growth forest specialists. And because of that, in California, they're increasingly affected and threatened by these large wildfires that convert continuous forest into these very different open landscapes. Because wildfires have become such an omnipresent threat in California and elsewhere, especially in the Western United States, the Forest Service has been using fuel reduction treatments to prevent wildfire. So they're actively removing undergrowth by either using controlled low-impact burns or by vegetation removal. That's great if it prevents large wildfires, but of course it also constitutes a form of disturbance of the forest habitat. And so we wanted to know how these fuel reduction treatments actually affect northern flying squirrel densities. And we had a huge data set to look into this question. It was 5 years of intensive live trapping and marking, and each year these people operated more than 1,400 traps. Anybody here who has worked with small mammals knows that this is an insane sampling effort. They were able to do that in an experimental forest where they could implement thinning and burning and have control units. And so here you can see a timeline. We have data from before these experimental fuel reduction treatments and data from after. And the one covariate we included here that we thought would affect northern flying squirrel density is canopy cover. Why did we include it? Well, it's important for squirrel movement, right? They're flying squirrels. They glide from tree to tree, so they need high canopy cover. And it is also reduced by fuel reduction treatments. And here you see I apologize, very pixelated map of our study area that shows you the amount of canopy cover after those fuel reduction treatments. And you can see dark green means canopy cover is high. So these dark green patches are controlled patches where no fuel reduction was implemented. And these lighter patches are patches where fuel reduction treatment was implemented, and therefore canopy cover was lower post-treatment. So we analyzed these data with the spatial capture recapture model with inhomogeneous densities. And we found that fuel reduction treatment did reduce average canopy cover from about 85% pre to about 70% post-treatment. And in pre-treatment years, canopy cover had no effect on northern flying squirrel density. They were uniformly distributed throughout the study area. But in post-treatment years, canopy cover did have a positive effect on density, meaning that in areas with remaining high canopy cover, squirrel density tended to be higher. Interestingly enough, though, when we looked at overall density or population size over the entire study area, that was not impacted by treatments. So the fuel reduction treatments led these animals to shift their distribution, but apparently did not affect their overall population. And so that's a nuance we would have missed had we not looked at changes in distribution of animals and only looked at changes in population size. So to summarize, spatial capture recapture is a framework that's much more data intensive than occupancy. You need spatial information, you need individual level information, but it can also do a lot more than occupancy or even a regular capture recapture models. It goes way beyond abundance. It can give information about spatial variation and density. It can be extended to look into space use and resource selection, or even connectivity for movement, spatial population dynamics and dispersal, and so forth. And with the increasing abilities of collecting individual level detection data with non-invasive genetics, for example, but also with better and better technologies such as camera traps and acoustic detectors, the use of spatial capture recapture is definitely also on the rise. So to give you a general outlook, I hope even focusing on just these two specific frameworks, I was able to give you an idea that the concept of hierarchical modeling is an incredibly flexible framework. Basically, even though there are all these different implementations of these models, there's always the same mold. You formulate a statistical model that describes your state, whether that's occupancy or abundance or whatever, that could be influenced by covariates. And then conditional on that state, you formulate a second statistical model that describes how your observations are generated, whether that's counts or whether that's species detections or individual detections. Again, the observations are conditioned on the latent true state. And within that general mold, the flexibility is almost endless. So for example, you can tailor this specific part, the observation part of your model, to the specific circumstances of your study. So for example, spatial capture recapture models have been extended to allow for the situation where you can't always fully identify individuals, where you may be only able to say, well, I know this is not individual B, but it could be individual A or C. It allows you to combine data as well. You can have different data streams that inform the same ecological state, but come from different observation processes. So that's, for example, done in integrated population models, and that allows you to estimate parameters better, can allow you to estimate entirely new parameters. And I've only shown you fairly simple state models, right? We've only looked at occurrence and density and how density varies. But there are also way more complex state models, such as multi-scale community occupancy modeling, where you not only look at how species are distributed, but also which species occur in which community. So an incredibly flexible and useful framework for anybody interested in studying wildlife. And with that, I would really like to give thanks to all the co-authors of the studies that I chose to present to you in part today. And I want to thank you for listening to me talk about statistics for so long.
Thank you,
And of
Rahel.
course, I'm happy to answer questions.
Thank you very much. That was excellent. I learned a lot. See, I definitely needed this class. So now I know more about this. So yeah, and I know other people in the program are more familiarized than me with that, but I've had students do some of that, and I've been trying to understand it better, but it was good to have an overview, and your studies are really interesting. And congratulations.
Thank you.
So I'm going to open for questions. It can be in English or Portuguese. Perguntas podem ser em português também. Aprendi a fazer no YouTube aqui, então temos mais de 40 visualizações no YouTube nesse momento e um chat aberto também. Então, lá e aqui. Perguntas do público, senão vou eu começar a perguntar. Estou tentando ver.
To digest. So
Yeah.
no problem.
No, but I'll have several. A geneticist will have many questions on that. So you were talking about the integrated model. We're interested in that because we're using eDNA, and fecal sampling in our field studies. So how common is this these days that you can, for example, integrate camera trapping with fecal DNA monitoring? And of course, a problem we've been having is that you can't necessarily get the same individuals, right? Or you don't know whether, like, you recognize cats using coat patterns with camera traps, and then you have their DNA profiles with scats. But we've been debating how to integrate, like with hair traps, to try to get DNA and camera images at the same time. But then if you use a hair trap with a lure, then you're going to bias your camera trapping effort, which assumes no attraction. So how do you see this being dealt with globally?
So I think it's a great question, and it's not always 100% straightforward. As you said, you can have two types of sampling happening in the same study area, and ideally you want to use both types of data to get at population size or density. But if you can tell based on code patterns who is who, and you can tell from the genetics who is you can't necessarily match the genetic data to the camera trap data. And there are multiple ways you can deal with that. You can either make the assumption, which it's not statistically correct, to say, well, we use those as two totally independent data sets and build a model that shares that state process. So it shares the density, but has two different observation models that both inform the state model. You can think of it like very simply, what you'll get out of it approximately will be the average density from the camera trap data and the scat data, and that'll hopefully be an overall less biased representation of density. But of course, those two data sets aren't independent because you're sampling the same individuals. There are more complicated models that can explicitly model the dependence of those two data sets. For example, because you have that spatial information, that's also what underlies these spatial capture recapture models for partial identity. So let's traps and a grid of hair snares or scat transects. Well, you know that the individuals only move over so much area. So if you get a hair snare or scat detection of an individual over here, and you get a camera trap detection of an individual way over there, they're unlikely to be the same individual. Whereas if you get them close together in space, they're more likely to be the same individual. And so because we retain that spatial information in the model can actually at least attempt to resolve who's who between those two data sets. That's the first part. And then your second part of the question was about using lure and thereby influencing the detection probability. That's actually not a problem because you can again account for, again, if you have some sites with lure and some without, because with spatial capture recapture, for example, now you're modeling your data in a spatially explicit manner. You can actually account for the fact that, oh, maybe at these cameras you applied lure on that day, and then on these other cameras you applied it at a different day, and just build that into your model.
Cool, that's reassuring.
Yeah.
So we can play with that some more.
Yeah.
Questions from other people? Perguntas, pessoal, para os alunos, os ecólogos do programa? Senão o geneticista vai continuar perguntando. Estou vendo quem está. Bom, fazer uma pergunta em português para o pessoal. Go ahead, Albica, dizendo só para eu go ahead. Então, vamos pensar em mais questões. Então, fazer em português, tá? Vamos ver se o pessoal se anima mais. Então, sobre a evolução desses métodos, né? Qual você acha que é o futuro em termos de incorporação? A gente também trabalha bastante aqui com eDNA, né? Então, você acha que isso vai tomar conta, assim, em termos de detecção? O quanto as pessoas já estão usando eDNA misturado com camera trapping e se isso você acha que é uma tendência ou é muito heterogêneo e também em termos de diferentes países, né? Onde é que você vê isso avançando mais?
Eu acho que está começando, com certeza, mas ainda tem muitos problemas específicos desse tipo de métodos para resolver. Por exemplo, se você coleta eDNA, é muito difícil determinar a escala temporal da presença da espécie que você está estudando, né? Sim, DNA não permanece na paisagem para sempre. Em alguns lugares, talvez nem permanece por muito tempo. Mas, por exemplo, trabalhando com iDNA, agora eu não sei a palavra em português,
You
leeches.
can say sanguessugas. Yeah, you can say it in English.
Sanguessugas,
You can
exatamente.
reply in English too.
Aí, uma sanguessuga fica com sangue por 8 meses. Então, se você detecta uma espécie numa sanguessuga, qual é o tempo, o ponto temporal que essa espécie estava presente nesse ponto de coleta? É muito difícil. E, e essas perguntas acho que vai demorar um pouco ainda para resolver, para como podemos modelar esse tipo de dado novo, visto que a coleta desses dados tem esses vieses, essas dificuldades muito específicas que vai demorar para resolver.
Uma boa resposta, porque a gente aproxima isso por outra abordagem, a questão molecular mesmo tem seus próprios problemas, né? Então, chegar em nível de espécie, em muitos lugares, para a maioria das comunidades ainda é um problema.
Uhum,
Chegar em nível de indivíduo, que é o necessário para dentro de uma adicional, ou seja, marcadores diferentes, têm que ser desenvolvidos. E também tem probabilidades de amplificação de funcionamento diferentes. Então, assim como tem probabilidade de detecção diferente de uma camera trap, também vai ter probabilidade de
uhum,
detecção diferentes de cada espécie e talvez até de indivíduos diferentes, de sexo.
uhum.
Então, tem toda essa parte de otimização do método de detecção.
Pois
Essa
é,
questão temporal é outra ainda, né? Dependendo do método. Então, realmente, acho que é bastante complexo para modelar isso. Talvez
é.
não aconteça.
Mas, por exemplo, a questão desses vários níveis de detecção, de probabilidade de detecção, você pode imaginar um modelo hierárquico com mais níveis. Eu agora falei de 2 níveis, né? O estado que você está interessado e a detecção. Mas você pode ter mais níveis, pode ser a presença da espécie, a presença da espécie na sua amostra, a habilidade de identificar a espécie na amostra. Então, pode ter mais níveis que descrevem todos esses níveis de identificação no processo de análise genético também. Isso eu já vi, alguma publicação que modelou todos esses processos.
Sim. É um problema que a gente tem muitas vezes em modelos, né, moleculares, é a variância, né? À medida que o modelo vai ficando mais complexo, aumenta a variância.
Uhum,
Eu imagino que o mesmo vai acontecer aí. Então, tem prós e contras, né? Ele fica mais inclusivo, mais flexível, mas a taxa de erro, devido ao aumento de variância, vai ser muito maior se for um modelo ultra complexo, com muitas camadas, né?
com certeza. Daí você vai ter que coletar muito mais dados para poder dizer algo interessante.
Exatamente, mas é muito interessante, extremamente relevante esse desenvolvimento metodológico. Perguntas, pessoal?
Eu
Walter,
tenho uma pergunta.
muito bem.
Por favor.
Tudo bem, gente? Bom dia. Posso fazer em português a pergunta?
Acho que
Pode,
pode.
por favor.
Obrigado pela fala, Rahel, muito legal. Tem alguma aderência assim com o que eu tenho feito, né, no laboratório com o pessoal, mas eu queria fazer uma pergunta relacionada com esses vários papers que têm saído atribuindo um nome para o conjunto de espécies que está ausente do pool de espécies de um local, que é a diversidade escura, dá para traduzir assim em português, talvez, que é a dark diversity e tal.
Uhum.
E eu queria saber o quanto que modelos hierárquicos parecidos com o que tu apresentaste para nós aqui estão sendo usados para essas estimativas, assim, porque tem muita discussão da importância desse componente da diversidade ausente, onde ela poderia estar, né? E obviamente tem uma questão de detecção também envolvida em estimativas de diversidade escura. Eu queria saber um pouquinho se estão usando esses modelos para isso, o que que tu acha sobre isso?
Bom, é uma pergunta muito boa. O problema é que nunca tem, não tem como provar a ausência, né? É o problema filosófico fundamental. É, mas eu acho que esses modelos têm um papel nessa questão de tentar determinar a ausência, quem está faltando num pool de espécies. Eu vi poucos papers que usam esses métodos para fazer justamente isso. Um ou dois, porque A parte legal é se você tem uma probabilidade de presença, você automaticamente tem probabilidade de ausência, né? Porque é já só um menos a probabilidade de presença. Então, se você consegue estimar um, você consegue estimar o outro também. E dentro da estatística bayesiana, você pode, por exemplo, ir para um lugar, fazer uma amostragem, você não detecta a espécie que você estava procurando. Então, como que você pode decidir se ela realmente está ausente? Aí você pode usar o conhecimento anterior, o que você achava antes de fazer a amostragem, o que você achava que era a probabilidade dessa espécie ainda estar no lugar, e combina isso com os dados que você coletou, que na verdade são um monte de zeros, né? Porque você nunca detectou a espécie. Então, você pode usar a estatística bayesiana, combinar o que você achava antes da amostragem, que era a probabilidade de presença, combinar isso com todos esses dados de não detecção e obter uma probabilidade de ausência. O que eu acho que é um método interessante para chegar mais próximo à verdade de ausência. Daí você poderia dizer, ok, se a probabilidade de ausência é mais do que 90%, a gente vai tomar a decisão que a espécie não está mais nesse lugar. Então, eu acho que esses modelos podem ter um papel nesse tipo de questão, mas eu conheço talvez 2 papers que usam esse método para tentar dizer algo sobre a ausência.
Não, legal, porque os que eu li até hoje tem muita metodologia da parte mais matemática relacionada com concorrências, né, com utilização de
Uhum.
dado de concorrência. E eu nunca vi, pelo menos eu não sabia nem que tinha algum que fazia isso mesmo para essa área. Porque, enfim, essa ideia da ausência verdadeira tem uma relação íntima com a questão de modelagem que chamam de nicho, mas enfim, modelagem de habitat, de ambiente, de nicho e tal. E tem enfim, tem um trabalho super antigo que tu deve conhecer do Mike Kearney, lá da Austrália, que ele discute lá em 2006 isso, que essas modelagens de distribuição, que ele é bem incisivo, assim, dizendo que não daria para chamar de modelagem de nicho, porque não tem nenhum mecanismo causal envolvido ali. É justamente isso da diversidade escura, né? Que se a espécie não é detectada naquele local, mas ela poderia estar lá por questão de nicho fundamental, essa ausência é
Uhum.
muito importante. Mas eu concordo, ela é mais probabilística do que real mesmo. Então, legal, muito legal.
Muito bem. Mais alguma pergunta, pessoal? No chat aqui no YouTube não veio pergunta. Mais alguém? Olhando aqui, não. Então, vamos encaminhar o encerramento. Rahel, muito obrigado. Foi excelente a palestra. Obrigado aí pela participação de todos. Essa palestra vai ficar gravada no canal do YouTube do programa. E quem quiser entrar em contato com a Rahel, fazer outras perguntas, também pode falar comigo, que eu repasso o e-mail dela, né? O professor Júlio está escrevendo no chat aqui, excellent talk, thanks, Rahel. Então, realmente foi muito boa, it was really excellent. E eu aprendi muito, acho que todos aprenderam, e acho que tem muitas opções aí de uso, de aplicação, pelo que eu conheço das pesquisas do pessoal aqui do programa, acho que muitos podem se beneficiar desse tipo de método e de contatos com a Rahel. Eu também divulguei essa palestra para outros programas de pós-graduação do Brasil, na verdade, para todos os programas, são 144 programas de pós-graduação na área de biodiversidade no Brasil, e eu mandei o anúncio e o link do YouTube para todos eles. Então, espero que mais pessoas tenham visto. Pelo que eu vi, teve 43 visualizações no YouTube aqui durante a palestra, que provavelmente vão aumentar à medida que as pessoas acessem a versão gravada, né? Então, mais gente vai poder assistir. Então, muito obrigado e mantemos contato. E, né, nos vemos no próximo seminário. Rahel, um grande abraço e muito obrigado. Parabéns.
Eu que agradeço. Obrigada pelo convite.
Até mais, então. Um abraço a todos.
Tchau.
Tchau.